episode 284 of the Stem Cell Podcast, Organ Chip Engineering, with Dr. Milica Radisic. Hello, everybody. This is Dr. Daylon James. I'm with my partner, Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge and stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Milica Radisic. She's from the University of Toronto, and she's on the podcast to talk about her research on organ chips that model cardiac disease. Very exciting, near and dear to both of our hearts. Very clever, pun intended. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, did you know that you can model arrhythmias and cardiomyocytes derived from human pluripotent stem cells? Watch Stem Cells On Demand webinar to learn how patient-derived and gene-edited human pluripotent stem cell lines can be used to model cardiac disease in vitro. Visit www.stemcell.com/cardio-webinar to learn more. We're going to start things off not with the cardiac paper, but with a paper coming from none other than Hans Clevers, the man himself, organoid aficionado, mastermind, maestro, whatever you want to call him. This is a Cell paper, pretty big deal. It's a developmental milestone, I suppose, for the human fetal pancreas, and they were able to identify a long-term in vitro expandable human fetal pancreas stem cell that can generate all three pancreatic cell lineages. And of course, we've talked about Hans Clevers a lot on the show. You know, he's been working on this area of study for a couple of decades now. First author here is Amanda Anderson-Rolfe. And you would think that something like this would have been figured out 2 decades ago, maybe, right? When Hans Clevers started working in this area. You know, it's almost like, you know, it seems like we've figured out how the pancreas develops, right? And apparently we haven't been able to do that or distill that into the organoid model. So this is really the focus here. You know, the pancreas has 3 different epithelial compartments, which actually derive from a common pancreatic progenitor: the acini, the ducts of the exocrine pancreas, and the endocrine islets of Langerhans, right? The acinar cells release the enzymes that break down food. Ductal cells make these channels to create carry enzymes to the gut. And then the endocrine cells produce hormones like insulin to control sugar levels in the blood. And of course, in the stem cell field, in the modern stem cell field, there's a huge interest in making those pancreatic beta cells for insulin production, right? And most pancreatic organoids, and I actually didn't know this up until now, only contained one type of these cells. But in this paper, they were able to create an organoid that contained the 3 major types of pancreatic cells, the ones that I described, right? So it's an organoid that contains all the cell types ideally found in the real pancreas. Of course, this is great for modeling development, modeling disease. So you can think of the variety of different applications here. And also, importantly, they're able to report the derivation of 18 human fetal pancreas organoid lines from gestational weeks 8 to 17 fetal pancreas samples. So this is great. That's a reflection on the reproducibility of this protocol. 4 of these lines, which are derived from 16 gestational week samples, generate the acinar, the ductal, and the endocrine lineage cells, while, get this, they're able to expand exponentially for more than 2 years under optimized culture conditions. So that's great. This is reproducible, it's a stable protocol. You can maintain these organoids for a long time. They contain all the lineages that you would expect to find in the fetal pancreas. And then, of course, they did the characterization. They did RNA-seq, single-cell RNA-seq, to identify rare LGR5 positive cells, pancreas, and also these fetal pancreas organoids as the root of this developmental hierarchy that turns into these 3 different types of cells, right? The other thing was, I think, the reflection on the power of these LGR5 positive organoid derived cells, because just a single LGR5 plus organoid derived cell could recapitulate the entire tripotency, the derivation of those acinar ductal and endocrine lineage cells. A single cell could recapitulate all of that in vitro. So it's pretty amazing. They found this unique type of stem cell that has the ability to turn into all different cell types of the pancreas. And I think, it opens a lot of doors, like I said, for modeling, for understanding development. And pardon me, I'm just surprised it took this long to get to this point. Yeah, I would have thought they would have figured this out when Hans first started, a couple of decades ago.
Yeah, that's it's just what you said at the beginning and end. This is something you're like, yeah, this is the thing. If I were to say, name the one thing to everybody, or like name the one thing that if you could just do it, wave a magic wand, I think a lot of people would say, let's just get a cell line, that is infinite pancreas in terms of unmet need, in terms of impact. And then it's like, yeah, if beggars were horses, right? So I think that this is something that a lot of people have gone after and given up on. We don't even talk about it as a holy grail anymore because I don't think everyone thought it was just not a thing. But of course, leave it to Hans Clevers, who I thought had moved on. He'd made every organoid known to humankind, and moved on to his commercial ventures. But now I'm thinking I want to look at the article metrics on this. This might have been his thing where he was like, you know what? I've run out of things to do with intestine. I'm going to go fix the pancreas for the whole field and join a company. And I wonder if this wasn't when he got to, because that's the other thing. It's not just like it was tough. I mean, this is a good model, I think. One thing to look at how they systematically approached it, to look at like the integrating the known quantity of LGR5, all that stuff. I think it's a good model for other organs that you didn't think you could do this for. I don't want to say the heart because that does seem impossible. But the other thing that you got to recognize is that over 2 years, that's insane. So the measure of proof that was necessary to really validate the claim, I think, demanded an effort like his and this and extensive review period. And I wonder if this wasn't the trigger for him. It was like, wow, we got to get this out there to save millions of lives and the economy generally. Um, and that's why he busted out. So either way, this is an amazing story.
Really great stuff. And something that you've mentioned previously on the show, this is also a reflection on the importance of fetal tissue. We like to focus so much on iPS derived cells, which are, of course, so powerful. And there are certain ethical advantages of using iPSCs over traditional fetal tissues, pluripotent tissues. But this is, again, an organoid type that was derived entirely from human fetal tissue. So, once again, just a reflection on the importance of that particular area. And who knows, maybe down the road there will be a human fetal cardiac organoid. Of course, it can't perfectly recapitulate the structural elements of the heart, but maybe Hans is working on that too.
I would not be surprised. That's like two on the list, right? Of impossible things. Um, Leave it to Hans. But yeah, I'm also glad you mentioned that fetal tissue demand and the one byproduct. I wonder, and I think they mentioned this. This is like, or they alluded to it perhaps, that there'll be like a demand for fetal tissue now created out of this. But I think that's the whole point of this, is making cells like HLA banking, et cetera. Yeah, you'll need to maybe set it up, but this it provides the possibility, right? And think about the pancreas, right? It's like these are patients that they could be treated modularly. I think the safety profile on this is great. Anyway, we could go on about this paper for a long time, but we're going to move on to a little segue here, the fetal. So I'm glad I mentioned that. This is a very straightforward story, kind of a resource, but I think showed some real disease relevance as well. And it's hematopoietic, of course. I love the blood. And this integrates, I think, the fetal with the adult, and it runs the gamut. And that's the idea, is that fetal cells are and developmentally embryonic cells and adult cells and youth and age, they all have like different profiles. And like in development in particular, they're geared towards creating the organism, right? And all the tissues and organs therein. But then as adulthood approaches, you get homeostasis with periods of growth or whatever, right? And this is particularly, I think, salient in the blood, because you have to meet these age appropriate demands, but also in a very dynamic landscape that has to balance the prioritization of like the generic oxygen transport function, but like also maintaining the blood supply, homeostasis, but also response, right? The innate, the adaptive immunity, and also tissue regeneration. It's increasingly becoming relevant or clear, the relevance of hematopoietic cells in coordinating inflammation, but also regenerative processes, right? So the blood, right? It's this dynamic organ. That's comprised of billions of cells of many types that are dynamic through age. And that's the idea here, is how these stem progenitor cell properties change over the lifetime. And this is exemplified, the dynamics of this, as I kind of alluded to already. Like if you look at fetal liver hematopoietic cells that have a much higher self-renewal potential than the adult, also like adult versus fetal globins. Also in like a pathological context, you talk about the myeloid bias, right? Age skewing of a lot of blood disorders, hemoglobinopathies, bone marrow failure disorders, leukemias in the older population. So single cell seq, right? You think haven't we done this already? But the problem is that while single cell seq lets us see really high resolution, you're not able to integrate as well. Although there's all these computational approaches that are really great. A single effort. There's been efforts with distinct ages, individual ages like fetal liver or cord blood or adult bone marrow, but a single comprehensive integrative analysis across the lifespan has not been done. Enter the pretty expansive big name group here that was led by corresponding authors last R. Grant Rowe and first Ho-Jun Lee, who were at the Dana-Farber, also Boston Children's, Harvard Med School, but a lot of co-authors on here, including David Liu, Aviv Regev, George Daly, and a bunch of other big names. So a comprehensive effort, I'm guessing, to recruit all the different samples. I think they had 26 different individual donors spanning in fetal liver, a bunch of those put through adulthood, early age to older individuals, and then deploying all the sequencing, of course, while you got people like Dr. Regev in there, et cetera. So bottom line, I mean, straightforward resources, a Nature Methods paper, although Nature Methods papers are very high impact. And the implementation, I think, is why I highlighted it. I love the blood. I love a resource. I'll definitely take a look at this data set. I'll have to beef up my computer in order to encompass all these cells. But the takeaways, which I thought were really important, were that they identified this, like, they functionally validated this fetal state by doing engraftment, which was nice. I think maybe important for review. But also more important for me was that they showed, in looking at like AML, acute myeloid leukemia, they used the profiles of that whole span and showed that if you had a signature that was utilizing the fetal program, it was a bad prognosis, which makes sense intuitively, but is important to demonstrate. It shows the disease relevance of the platform. And I think the key is having a high resolution data set like this with a lot of targets, is that you may be able to dip in terms of therapeutic extensions of the data set here, is you might be able to look in to that fetal program and maybe target it, not only get like a diagnostic prognosis, a risk profile, but also maybe have a patient-specific therapeutic. So I love a blood story, but I think this also deserves it as like a new standard for comprehensive, like I said, integrative data set, taking into mind that hematopoiesis is a very dynamic process.
Yeah, this is a real who's who of molecular and cell biologists on this paper. Of course, you got David Liu, you got Stella Chow, Aviv Regev, you got one and only George Daly. Last author here is Grant Rowe. This is a comprehensive effort across many of the different institutes at Harvard and the Boston area, in particular Dana-Farber, which is, in my mind, the world's perhaps one of the top 2 or 3 leading cancer institutes, cancer research institutes in the entire world. And they have the resources, they have the patient population to be able to do something like this. And of course, this is uniquely enabled by what Aviv is able to do, even though she's not directly at Harvard anymore. She's at Genentech and has other affiliations. No doubt this paper has been in the works for a long time. So really great reflection on the collaborative nature of the folks up there, although it's a little bit too cold for my interests these days.
Yeah, you said a hit list there, and I don't want to be cynical, but I think a lot of what pushed this through to Nature Methods, as like in that it was a resource. It was important, I think, but those big names pushed it through with a relatively abbreviated review for the Nature journals of less than a year. Unlike the paper that I'm coming at you with after yours, which was languishing for a couple years. Not a Harvard story, not coincidentally. Happened in my neck of the woods, MSK, and they put it in review for 2 years. Arun. All right, I'm going to stop and let you get to your roundup story.
Seems very personal for you, but no, we all feel the pain, man. These reviews, these papers take way, way too long to get out. But that's a discussion for another day. This is moving on to another big high profile paper in Science. This is in the topic of mechanotransduction, something that's gotten a lot of attention recently. Of course, the Nobel Prize for, I forget what it was, but it was for mechanotransduction within the last few years. I think it was Artem Patapoutian, right, who helped discover those piezo channels. And that's exactly what this paper is about. It's about the piezo channels, and it's about how piezo dependent mechanosensing is essential for intestinal stem cell fate and maintenance. We're talking about Hans Cleaver's, we're talking about the intestine here as well, but different approach, I guess, a different angle to things, and figuring out how mechanotransduction is really critical for stem cell fate and decision and maintenance in the gut. So, tissue turnover, regeneration, we talk about this kind of stuff all the time here on the show, right? We've talked about it in the context of the axolotl, we talked about it in the sea anemone, we've talked about it in so many different contexts. And that's those functions of tissue regeneration renewal are, of course, contributed to by stem cells. And the intestinal epithelium, in particular, has this amazing regenerative and renewal capacity and a pretty powerful ability to regenerate after an acute or chronic injury. The mammalian intestinal stem cells, as Hans is very familiar with, are located in this pocket like structure that we've talked about before on the show called the crypt. And they respond to signals in that microenvironment of the crypt. A really beautiful structure. I'm sure everybody's seen it by now. They respond to these signals and maintain that balance of self renewal, proliferation, lineage commitment. And you need this in the intestine. It's such a rapidly turning over tissue, right? So you got to figure out how this works. And we've done, not us, but many people across the world have done decades and decades of work in this area and figuring out how the intestinal stem cell populations are committed, differentiate, renew, and so on. But this idea of mechanotransduction hasn't always been examined in this region, in this crypt, and in particular how physical cues might influence intestinal stem cell behavior. They're trying to figure out here how ISCs, intestinal stem cells, integrate the mechanical signals from their niche in vivo. That's the thing here. You can do a lot of these studies ex vivo, sort of like bioengineered, tissue engineered approaches, sort of like Matthias Lutov can do, right? But figuring out how these mechanotransduction signals and properties influence intestinal stem cell behavior in the crypt, in the native crypt in vivo, that's a tough question to answer. And that's what they're doing here. And the Tae Kim lab, first author here is Miriam Baghdadi. And so that's what they did. They wanted to define That's what they wanted to do. They defined the roles of the piezo channels in intestinal stem cells. They performed in vivo conditional genetic ablation of the piezo genes, and if I haven't mentioned by now, those piezo genes are thought to be the hallmark canonical genes for mechanotransduction in various tissue types, right? And they found that double knockout piezo-1 and piezo-2 mice exhibit stem cell loss, which leads to a rapid lethality. So you need those piezo channels to properly regulate intestinal stem cell function. And without their function in the intestine, those animals die. They did a bunch of single cell transcriptomic analysis of the intestinal crypts isolated from these piezo knockout mice and show that the ISCs lose their stemness and acquire a really highly proliferative phenotype that leads to their depletion ultimately, and that presumably leads to the lethality of the animals. So they show that these pre existing multipotent translate amplifying cells can differentiate preferentially into absorbent progenitors, which leads to secretory cell deficiency. They did a bunch of, like I said, single cell RNA sequencing combined with ex vivo three-dimensional organoid culture to validate the function of these piezo channels in regulating intestinal stem cell activity. And they demonstrated that notch hyperactivation and Wnt signaling inhibition uh, when this piezo activity was blocked. So there was a counter effect there. They did atomic force microscopy, as you might expect, with studies of mechanotransduction to measure the basement membrane stiffness in mouse small intestines and show that these ISCs experienced a more rigid microenvironment at the bottom of the crypt compared with the cells actually at the top of the crypt. I mean, I can't imagine how difficult that must have been to do atomic force microscopy in vivo and in native architecture. That's pretty exceptional. And then they also did some organoid monolayer culture, 2D and 3D, that were cultured on bioengineered substrates of different rigidities, showing that the stiffness can indeed regulate intestinal stem cell behavior through piezo signaling. So it's not a brand new concept. Of course, mechanotransduction has been around for a long while. Piezo channels have been talked about forever. We've been talking about them here on the show in various contexts. But I think the power of this study is figuring out how those piezo channels, those canonical mechanotransduction channels, actually influence intestinal stem cell behavior in the native niche compartment, in that crypt. And doing that in vivo is exceptionally challenging, and being able to interrogate that is challenging. So, huge props to these folks for figuring out this fundamental mechanism of how intestinal stem cells are regulated by mechanotransduction.
Yeah, they did so much. This is a lot of work. I guess, yeah, not a brand new conceptually, but I think it really is another brick in the wall highlighting the importance of mechanotransduction. I mean, we're going to go to they're going to have a whole conference about mechanotransduction and its role in like lineage specification, not to mention homeostasis and like organ architecture. And you read papers like this, and it gets me to thinking, at least, about how critical, like, okay, I'll say this, like, I think a really important part of this study, the only reason they were able to do it is because intestinal organoids have this really unique way of recapitulating the architecture. I mean, that seems obvious, but I think if you extend that idea to the field in general, I've got a hot take. And if you guys disagree out there listening, please leave a comment. We need you guys to weigh in on this. Tell me what you think. I think we got to have a more subtle and refined classification of what we call organoids, because I think a lot of what's out there is maybe an organ-specific spheroid. In structure and shape, I think at least recalls at the cell level some of these mechanical, structural relationships that you see in the native tissue during differentiation, I think are really key. If you look at this, it seems like it's really key. Right. So I think that we need to qualify organoid maybe with a little bit more stringency or at least determine whether the distribution of cells and the overarching structure is either consonant or dissonant with the native architecture, because it's a key variable, clearly.
Yeah, those concepts of tissue architecture are, of course, imperfectly recapitulated in the organoid. That's why it's an oid, right? It's not an organ. But that's, we're not even talking about two-dimensional cultures, right? Which is a complete artifact. We grow these things on, very stiff plastic in polystyrene, and we expect to understand basic mechanisms of molecular biology by growing these cells in plastic that's nowhere near to what they are grown in vivo. And of course, I'm hating for sure, but we've been able to glean numerous insights over the years by doing that, growing these cells in simplified formats. But we just have to keep that in mind, that everything that we do is with a caveat. And like what we've said a million times, we work on models, right? And some models are useful, but all models are wrong.
That's right. But this one, I'd like this one. The oldie but goodie, intestinal organoids, the first oid. I think it's less wrong than the others. And here they're bringing out even more insight from this old dog. So glad to see it. And clearly, there's more to be learned, I think, across the field, but specifically here still in intestinal organoids. The original gangster. Moving on. I got a story here that's it's a cancer story. It's a cancer story, like it's not, there's no stem cells here really at all. But even the authors, I think, highlight the relevance to our field, stem cells and regenerative medicine, in the discussion. And I agree. I think this is really conceptually a bombshell, even though it's like intuitively there. And I alluded to with my rage how long this was languishing in review, but it's like really deep. So I don't know. Maybe they just put so many targets up there that needed to be tied up, because this is a really complete story from Thomas Tamalad at my neighboring institute here, MSK. Yeah, and it's about cancer, right? So cancer, the incidence sharply increases in the 50s and 60s, right? As you get the accumulation of these somatic mutations in adult stem cells, right? And that's the key, because the stem cell, these adult stem cells, they're the cell of origin for most, if not, I don't want to say all, but most cancer cell types. But the converse relationship is also there, which is that as you age, so you become more at risk for cancer as those somatic mutations pile up, but also as you age, the fitness, the number and the fitness of these stem and progenitor cells declines, right? And who knows if there's like a teleological reason for that, or evolutionarily, funneled in balance or equilibrium there. But whatever the cause, it serves to counterbalance the incidence of cancer, to the effect that cancer depletes, the incidence of cancer depletes rapidly, like in your 80s. If you make it to past your 70s, you don't get cancer, like your risk profile goes way down. And these aging associated changes, they're conserved in mice. Who knew? Mice are a good model for aging. I don't know. May have a bone to pick with that, but at least theoretically. And the math is that like after 2 years of age, these aging associated changes begin to manifest, and that age corresponds to about 65 to 70 years of age in humans. So I guess the math lines up. But either way, you see at the cellular level, you see these things starting to manifest. So this is a story about lung adenocarcinoma, which is 7% of all cancer mortality worldwide. Mean diagnosis 71 years. And as you age, the incidence increases, right? In smokers and non-smokers. It's just like this pile up of somatic mutations. I guess environmental exposure certainly has something to do with it, even the absence of smoke. But as I said, the incidence declines after 80 to 85 years of age, which you wouldn't expect, right? So the thinking is that it's this mechanism that accounts for it. Those stem cells that they don't have it in them to make a cancer. They just give up. And looking at this, the investigators in the Tamala lab, they employed this model, right, that it causes genetic mutation in these alveolar type 2 cells of the lung, which are the cell of origin for most lung adenocarcinoma. And the way it works, they have this activation of KRAS and a deletion of P53 in one shot. They call it this KP model. And it causes this de novo incidence of cancer, right? So using this, they showed, and this was like just the overarching idea, right? Intuition. They showed that indeed, looking at older mice, you get versus young, old versus young was the comp, and they showed that lung cancer initiation and progression was less, right? There was less initiation, and then they went deep on the mechanism. They showed that this was because you had this age-associated induction of NUPR1, which is a transcription factor, and that targets lipocalin-2, which also was induced in the aged mice. And then that leads to an iron insufficiency in these aged cells. And if you either inactivate that axis, the NRPR1 and lipocalin, or if you supplement with iron, you rescue the stemness of those cells, and then you get tumors. I mean, you can keep your iron for me. But the other thing also, which was interesting, is if you knock out that axis again, the NUPR1 lipocalin axis in young mice, it's bad for those AT cells because they induce ferroptosis, I guess, because it's like they're hyper iron. I don't know. Then they go in the DNA hypomethylation to show what's driving the NUPR1. And fundamentally, I think the takeaway is this intersection of iron insufficiency, which I would have never had on my card, leading to loss of stemness. And they're showing that you can modulate it, right, with just like supplementing with iron, very low. It makes me wonder about those anemics out there, maybe they got the edge, or maybe they're getting more cancer. I don't know. But the relevance, I think, was highlighted even by the authors, that this may be something that could be deployed not just for like cancer prevention, but also is something that we may want to keep our eye on in the context of regenerative medicine. I don't know, maybe in like reprogramming cells from older patients versus young, or just like basic, like, hey, let's try and supplement with iron to increase the stemness during our diffs. I don't know. But certainly something that raises a flag, I think, for a lot of people that may want to look into their cultures, or it may explain some things mechanistically. And finally, yeah, I think it was important in terms of showing where we should marshal our resources, showing that younger patients who develop cancer are at greater risk. And I think that's a fundamental change in our thinking, like, or at least accounting for that in a deliberate way, is that, oh no, a young patient with cancer is likely going to have a worse prognosis because of the increased stemness. And I think that's something that didn't occur to me like a priori before reading this.
Yeah, you always think about cancer as a disease of the old. And this is sort of turning that concept and that notion on its head. Kind of scary to think about that some of those initial sparks of cancer might be starting to accumulate now, or in even younger kids. And it's not until later on that you finally see the manifestation of that mutation, or kind of a scary thought to think about. But perhaps it can reframe our concept of cancer prevention or cancer early diagnosis. And like you're saying, perhaps target younger individuals and keep a closer eye on their propensity to develop cancer. And we've talked about tangentially this concept of a chip as well, this clonal hematopoiesis, which is a similar concept, right? You're accumulating mutations in hematopoietic stem cells, and that's ultimately associated with downstream phenotypic manifestations of various forms of disease, like cardiovascular disease, whatever. But same idea in a way, right? You have these mutations, you have things that happen earlier on in your life, and it's not until later on that you're seeing those manifestations. But a lot to think about here. I really like the model system here, but yeah, I think a lot of unique food for thought in this particular study.
Yes, food for thought. I like your first call, which was scary, which I was reading this in a cold sweat as I round the corner on 50. and just thinking of all the bad choices I've made in my life. Oh, Arun, for goodness sake, God willing, I dodged all those bullets, but at the very least, you know, we can get some extra focus on me as a young patient. I don't know what I'm doing here. I'm like manifesting cancer for myself. Arun, please, can we
Why would
start
you do
over?
that? Why would you do that, man? This is very dark.
Oh God, can we go to the B-roll? Anyway, moving on. We're gonna cure cancer anyway. I'm gonna be optimistic about that. I'm back in a cold sweat. Yikes! I'm gonna have to cool off because we got an interview in just a minute with Milica, and we're not going to talk about cancer. Thanks God for that. But we're going to talk about more delightful, optimistic things that are giving us hope for the future of cardiovascular disease treatment and just really cool organ on chip stuff that you guys are all going to want to hear. Before we get there, I got a quick message from Stem Cell Technologies. Do you work with human pluripotent stem cell derived cardiomyocytes? You should use perhaps stem cells, stem diff cardiomyocyte media and supplements to differentiate, enrich, expand, and cryopreserve functional HPSC-derived cardiomyocytes. Stem diff cardiomyocyte media is compatible with human embryonic stem and induced pluripotent stem cells, and the resulting cardiomyocytes can be used for disease modeling, drug discovery, and cardiotoxicity screening. You can learn more at www.stemcell.com/stemdiff-cardio. All right, everybody. Today joining us, we have Dr. Milica Radisic from University of Toronto, where she is professor and Canada Research Chair. The Radisic Lab focuses on organ-on-a-chip engineering to mimic physiology, the heart, kidney, and vasculature for the purpose of modeling human disease and discovering better and more effective drugs. They also synthesize new biodegradable and biocompatible polymers based on small molecules found in the human body. Milica, thank you so much for joining us on the show today.
Thank you so much for having me, Dalen and Arun. It's really a pleasure to be here.
Well, I must say the pleasure is ours. Not often that we have bioengineers on the show. I think we could use a lot more of that insight, given where we're going and how quickly in our field. Talking about organ on a chip, it's kind of like a catch-all at this point for this burgeoning science of interfacing the engineering component and the biology, right? The cells. So let's start by having you give your own, in your own words, overview of the research focus in your lab.
My research focuses on basically two broad areas, and you summarized it pretty well. One area is organ-on-a-chip engineering, and the other area is in instructive biomaterials. And my favorite organ is the heart, and I usually say it's the most important organ because life starts and ends with it. And of course, every scientist thinks that the organ they're studying is the most important. But we have a particular challenge with trying to reconstruct heart tissue. And when we say heart tissue, we mean the heart muscle. It is actually scientifically called the myocardium. And it's made up of beating heart cells called cardiomyocytes. And our challenge is that we cannot expand them when we harvest them from the human body. That's a really fundamental challenge because they don't really proliferate and they don't divide in adults. Shortly after birth, they stop dividing. And that's a fundamental challenge. Practical challenge is who would be willing to give you biopsy of their heart to make more heart tissue? This really doesn't get done. Biopsies of the heart are sometimes taken, very rarely, in people who have certain cardiac conditions. And usually those cells are not good. You will not be using them to basically try to make more healthy heart tissue. And the only reliable way to get cardiomyocytes, these beating heart cells, is from stem cells, from pluripotent stem cells, which means either induced pluripotent stem cells or human embryonic stem cells. And that's the connection of my research to stem cells. If we want to make heart on a chip, we gotta use stem cells. If we want to create engineered tissues with instructive biomaterials, we gotta use stem cells.
Yeah, well, I got to tell you, my favorite organ is the heart too. As listeners of this show definitely know, also my favorite cell type happens to be the iPS cardiomyocyte, and it's always fun chatting with a fellow iPS cardiomyocyte biologist, on the show. We've talked about the pros and cons of the cell type. How, like you said, it's the only way to mass produce patient-specific cardiac cells, and there are caveats associated with these cells. They're immature. That's the first thing that everybody comes up with, immature in comparison to the real adult human cardiomyocyte. But from your perspective as a cardiac bioengineer, what would you say is their greatest strength? And is it worth overlooking some of those immaturity issues for now, even though we are definitely making a lot of progress in that realm? So talk to us more about the iPS cardiomyocyte.
Well, so I'm old enough that I've started doing science in the last century, right? In the 90s. And back then, if you remember, or if you were reading papers, really the only source of cardiomyocytes were animals, right? We had to harvest them from neonatal rats usually. And that's, I would say, quite brutal. So we've gone a long, long way with Yamanaka's Nobel Prize recognized discovery of induced pluripotent stem cells. Now we can harvest in a very non-invasive way, either, for example, through blood donation, cells from any human. We can reprogram them and we can create pluripotent stem cells. And there are beautiful differentiation protocols developed by pioneers such as, for example, Gordon Keller. Sean Palachuk has a great protocol. There is a Burridge protocol. Stem cell technology sells a kit for cardiomyocyte differentiation. So now you can go from iPS cell to cardiomyocyte. You don't have to harvest it from animals. Back in those days when we were harvesting them from animals, they were also slightly immature, right? We were harvesting them from neonatal pups because adult cardiomyocytes from adult rat hearts, as I mentioned, they're terminally differentiated. In all mammals, we just can't make more. So we had to basically use immature cells from neonatal pups. And I'm so happy we don't have to do that anymore. And since we got our hands on iPS cells and human embryonic stem cells, but mostly iPS cells, our use of animals in research has profoundly, profoundly decreased. We simply don't do that anymore. So it's been years and years and years that we haven't sacrificed a single animal for this purpose. I would say that's a huge win. It's a huge win for ethical research and for reduction of animal numbers in research in general. They are, when they come out of differentiation protocols, iPS cardiomyocytes are immature. But over the past, I would say 10 years or so, there have been wonderful protocols published to enable maturation. And I would say they generally fall into several categories. One of them is biophysical stimulation. You could electrically stimulate them or mechanically in a tissue structure, and you would enhance maturation. Another group is basically metabolic maturation, and that's very popular right now, selecting the right type of culture media that has, for example, a lot of fatty acids, where you are trying to make cardiomyocyte switch to that fatty acid metabolism that's characteristic of adult cardiomyocytes. And the last approach is just simply growing them for a long time, right? If you have a long-term culture, you will be able to induce some maturation. And all of these protocols stop short of full terminal differentiation, like in an adult human heart. But that is probably not necessary because it's been very well established that tissues that are made out of these cells are able to recapitulate a lot of drug responses. And people have tested many groups around the world, not just our group or not just the company that we started, all the drugs from SIPA panel. SIPA stands for Comprehensive in vitro pro-arrhythmia assay, where there is this hope that we could use tissue preparations together with in silico modeling, but tissue preparations that come from IPS, basically derived cardiomyocytes, and we could predict what is the probability that a certain new molecule could cause arrhythmia in a human. And for that, for validation of your preparations, there is a recommended set of 40 drugs that are characterized as low, medium, and high pro-arrhythmia risk. So many preparations based on iPS cardiomyocytes have passed through this testing, and people have demonstrated they're really good in catching these basically subtle differences that could set you up for very serious arrhythmia. And that's why I would say, like, I think the tools are pretty good right now. We don't have to have full terminal differentiation to use them in drug discovery or even in disease modeling. In terms of transplantation, the jury is out there, right? Like the cardiac transplantation of cardiomyocytes derived from iPS cells is really not routinely done in humans, right? There are some desires to start clinical trials. There is a clinical trial in Germany that is run by Wolfram Zimmermann, where they're making tissues from stem cell iPS-derived cardiomyocytes. They're putting them back into patient hearts. But the jury is still out there, what would work best. And some people believe that you really don't need terminal differentiation for that application either, because that means the cell would be very sensitive to hypoxia. And obviously, patient hearts, failing hearts, have unfavorable environment in which you are transplanting cells. So having slightly immature cells could also make them more likely to survive in this transplantation setting.
Hmm. Yeah, I mean, I think everyone gets this. It's such a focal point of not just cardiac biology and stem cells, but like since the beginning, embryonic stem cells has been in large part about the heart, spinal cord, heart. So it is. It's understandable that it's the focus is always on that transplantation. But as you described there, and really only a fraction of the amazing insights that have been gleaned from using not just induced pluripotent stem cells, but just using the model, using these stem cell based in vitro and or transplant models to just understand these systems during this developmental and or pathophysiological context. And you yourself have made those insights in ways that like you wouldn't have expected, right? And as you're alluding to there, it matters less whether or not you reach the end point that everyone has in mind here, transplanting the cell in there, but all the tools you develop along the way and how those are applied clinically in part, right? You had this paper that I remember shortlisting for the roundup in cell stem cell. You mentioned Gordon Keller, Dr. Keller, the legend, that was there on there with you as well. But you were the lead, and it was extending the observations that have been made about this beneficial macrophage activity post neonatal cardiac injury in rodents into the heart on a chip platform. Like that is what I think is so brilliant, is that you can take these ideas from model systems that may be tenuous, or maybe someone will say that lacks clinical relevance, and you can say, no, look here, we have a system that can prove the principle to a threshold where we can move forward on this. And those are the questions that need answering, and those are the strides that are being made. I mean, what I loved about it was not just the utility. In this case, it was really beneficial, that like improving the perfusion and function of cardiac tissue. I loved how you elevated the yolk sac macrophage, right? The primitive hematopoietic derivative that everyone sneers at, but is really plentiful and practical to differentiate, right? So I love that this is something where, again, it's like this idea of like, yeah, okay, transplant, that's the holy grail, but look at this. We got these macrophage yolk sac primitive, they're right there. We could get infinite. And we have a way that can move the needle, can make it better. Maybe we can move that into patients post-MI. Like, who knows what the application is, but the insight is there. Tell us more about that study and how you hope to extend the results to future basic or clinical studies.
Yeah, I think that's a really exciting paper that we published this year in Cell Stem Cell, and it's a collaboration with Slava Appelman, who is here in Toronto, in Toronto General Research Institute, and also collaboration with Gordon Keller, who definitely is a legend of stem cell-directed differentiation protocols. And there are a lot of things about human body, human tissues, and human organs that we still don't know. Despite such tremendous progress in the second half of 20th century and through the 21st century, there are a lot of things we don't know. And Slava Appelman, while he was doing his postdoc with his team of researchers, his supervisor, and other students and postdocs, basically they identified that there are specialized macrophages in tissues that are kind of in the heart. They're called resident macrophages. And they're unique because they contribute to homeostasis. And a lot of papers have been published exclusively using rodent models that show that these macrophages are critical for the ability of the heart to regenerate. So if you injure a neonatal heart, and obviously this is done only in rodent models, within a couple of days after birth, this heart can fully regenerate. And some people believe, like, I should say heart muscle, just to clarify. And some people believe, well, if we can figure out what's going on here, then we could figure out a way to regenerate the human heart that is aging or injured by myocardial infarction or something like that. And so it's been demonstrated in a lot of papers that these resident macrophages are very important for regeneration and this regeneration ability of neonatal hearts. They're very important for angiogenesis. But the problem is in human hearts, we kind of don't even know their markers. We don't really know if this really happens in human hearts. And how would you even do this experiment? It's not possible. Like, there's no way you could do this kind of experiment in a human, right? You can't ablate resident macrophages in a human baby and see. It's completely impossible, right? And so the engineered tissues heart on a chip that we are making, where we are now in a very controlled way putting multiple different cell populations together, helps us understand the contribution of each cell type to tissue homeostasis, to the tissue state, right? It helps us figure out what are really the markers of these resident macrophages. How do they program? That's something that Slava was really pioneering, is basically, if all of these resident macrophages arise from the yolk sac, it means all of our tissues during embryonic development are seeded with the same cell type. But with time, they change, and now they become resident, let's say resident cardiac or resident some other tissue. And as they're changing their transcriptional profile, their gene expression changes. But we don't know how and why and what are the mechanisms and what's the end game, what's the end state. We don't have that information in a human. And through these tissue preparations that we're building, it's possible to study that. So this year, together, we have two papers, one in Nature Cardiovascular Research that was more, basically, it was a BioWire platform. And we published on this BioWire platform back in 2019 in Cell. And it's like a string of cardiac muscle that's made out of iPS-derived cardiomyocytes, some fibroblasts, and we spiked in some macrophages. And we were trying to study, does the tissue get better when you put in macrophages? And the answer is yes, the tissue beats better and it's healthier. There is less signs of death. And Slava's team led this work and in a collaborative effort. And basically they found that there is a lot of phagocytosis going up. These kind of primitive macrophages that came out of Gordon's differentiation protocol can really clean up this environment. They make it healthy for all other cells. And while they're chewing up stuff, they get programmed to the resin macrophage fate. And then we had a paper in Cell Stem Cell that my group led. Again, it was collaborative effort with the same group of scientists, where we demonstrated they're critically important for stabilizing vasculature in engineered heart tissue. Like, we could never get really nice vasculature, microvasculature in engineered heart tissue. And somehow this environment is so unhealthy, it's pro-inflammatory. We like to think that tissue engineering is like development, but it's not, because you're taking cells that are not really in their natural kind of state. You're mixing individual cells, they're a little bit unhappy, some of them are dying. That kind of triggers some inflammatory responses. And if you are able, and obviously if there is inflammatory response, you're not going to have stable vasculature. And when we spiked in these primitive macrophages, they're really a game changer. But it was a very interesting crosstalk between the cells, right? It's not like, we found that the main mechanism is not really macrophage talking to endothelial cell. You would think like, okay, macrophage, stem cell-derived macrophage is going to talk to endothelial cell and it's going to tell it, okay, be stable now, stabilize the vasculature. No, macrophages were talking to stromal cells, and stromal cells were becoming more pro-angiogenic, and that they were communicating those angiogenesis signals to endothelial cells. So that just tells you there is the reason why there are so many different cell types in our body. And with single cell sequencing now from the Human Stem Cell Atlas, we can learn a lot. And then basically there is this feedback loop. You can test your hypothesis in the engineered heart tissue, learn from what's published in Stem Cell Atlas, and that's how you push this boundary of knowledge.
Yeah, I think that those papers are a nice reflection of a number of things. One, the power of co-culture systems, right? The power of these auxiliary cell types enhancing, improved vascularization or maturation of the target cells, like the cardiovascular cells in your situation. But also the power of, it's a reflection of the power of the organ on chip system, which of course your lab has been pioneering for a number of years. And I've dabbled a little bit in the heart on chip field. We had a paper published this year in Lab on Chip showing how, active biomechanical stretch fluid flow can improve maturation of cardiomyocytes and endothelial cells on a very simple 2-channel system. But, one of the issues that we had, and I think a lot of other groups in this field run into, is the right substrate for actually growing the cells on the chips. Traditionally things like PDMS have been used with pros and cons. And when it comes to chip stability, there can be some issues, right? Could you talk more about the biomaterials used for organ chip culture and what's the state of the art in that regard?
Thanks so much for that question. Yeah, this is something also that was a really active area of research in my lab. And when we think about PDMS, polydimethylsiloxane, it was a revolution in the 90s, basically, when George Whiteside's lab figured out soft lithography, right? So all of these techniques, they come out from electronics industry, where if you open any of your devices right now, there are chips inside, and they're really microstructured. And there was basically a race how to put more and more and more on a smaller footprint. And usually these are metal pieces, but you have to process them in the clean room, and you get that precision to the micrometer scale of and even nanometer scale of the structures that they're building. So for a long time, people were using these hard silicon substrates, and they don't have, there is no E at the end, right? And so then a group around George Whiteside figured out, well, you could do this with a soft material, PDMS, which we call silicon with an E, right? And you could really shape it in different shapes at the micrometer scale. And that was a revolution, really a revolution that enabled microfluidics, enabled microcontact printing, enabled the creation of substrates of defined shape. And it's still a go-to material for a lot of applications, partly because we have tools. Almost every university now, probably not say every university, but basically every major city at least has one of these facilities, right? There could be one university in your town that has a microfabrication facility that anybody can use, right? It is now possible to even 3D print PDMS with a 3D printer. If you think about SUA masters, it's now possible to direct write them. You don't have to order your kind of masks on transparency or chromium and then do all the steps in the clean room. You could just 3D print directly SUA master from which you will cast your PDMS down. So I don't want to like say PDMS is a bad material. There is still a lot of use for it, especially in prototyping. But some limitations for widespread use have arisen. So another nice feature of PDMS, it's gas permeable. So gases can diffuse through it, which means if you have bubbles, you could find a way to get rid of them. Your cultures can breathe through PDMS, right? There will be oxygen exchange. But the limitations are really, well, it absorbs small molecules and small hydrophobic molecules. And if you want to make substrates for drug testing, that's a problem. Another problem is it is actually kind of expensive. As you start scaling up and you make more and more and more devices, then you realize, wow, we're spending so much money just on PDMS. And then the more devices you make, you realize this actually takes a long time because you have to kind of pour PDMS over your master mold. You have to wait for it to cure. You have to bake it in the oven. It's hours, right, for each device, which is okay if you're one graduate student trying to make a handful of devices. You can get some undergrads to help you. But if you're thinking about mainstream and now you want kind of everybody in the world to use some kind of 3D tissue, then you need tens of thousands of devices. You might need millions of devices if this really becomes a mainstream. And that's where PDMS cannot do anymore. You cannot process it that fast. And so industry that figured out how to process things fast is basically plastics processing industry, right? And I know plastics is not good right now, right? If you think about tissue culture polystyrene, and there are many other polymers that you could process by hot embossing or injection molding. And then if you go all the way to injection molding, that's really seconds for each piece, right? But the problem with plastics, well, it's too stiff, right? PDMS, you kind of bulk Young's modulus is about 1 megapascal. So you could make it a bit softer. You could play with dimensions, kind of get the stiffness that you need. And but with plastics, it's not possible, right? Because that's really as hard as a rock to the cell. Another advantage of plastics, like tissue culture polystyrene, it could be rendered very non-absorbent, right? You could coat it with different coatings to really prevent any adhesion of molecules or cells. So we believe really the way to go is multi-material processing, where a bulk of your device could be made out of something that is like tissue culture polystyrene that you could create quickly by injection molding or hot embossing. Hot embossing is less fast than injection molding. There are also materials like PMMA that you could micromill or polycarbonate. And that could be like bulk of your device. But the cell contacting parts, then you have to embed either hydrogel or elastic polymers, right? There is this overarching kind of approach or hypothesis that if you want to make a healthy tissue, you got to match the Young's modulus to whatever that tissue experiences in the body, right? And if you think about it, it could go from 1 kilopascal for brain to basically megapascal for bone, and muscle tissues that make up muscles, they're like from 10 to hundreds of kilopascals. So you got to make sure that the cell is contacting material that's softer, and then you have to embed them in some kind of hydrogel, or what we do is we use elastomers that we can 3D print or that we can deposit by 3D stamping into these kind of plastic holders that make up the bioreactor. And so far that worked pretty well, right? And going forward, really, sometimes people ask me, what do you think is success of organ chip engineering? And I think is becoming mainstream, right? It really, 3D tissues can help you answer certain questions, and everybody should be able to access those assays. It shouldn't be reserved for like special labs. And I can see some signs that the field is moving, our field, cardiac, is moving that way. A lot of labs are already able to implement cultivation of like a 3D string of cardiac muscle between two PDMS posts or in like a microfluidic channel. It could be just a string of cardiac muscle that contains elongated cardiomyocytes. So a lot of labs are going that way already. And then the big question for us as cell culture scientists, what do we do with all the plastic waste, right? What do we do even with all the polystyrene tissue culture plates that we are using right now? You know, some of them get recycled. Some universities and some institutions have programs for those, but some don't. And I think ultimately we got to collectively, working with polymer science colleagues, move towards making those devices also fully recyclable, biodegradable, and also relying on sustainable sources. Right now, basically, it's petroleum sources that are used to make these devices. So those are my kind of full circle thoughts about what are the good materials for production of organ-on-a-chip devices.
Wow, what a synthesis. I love how I mean, that's why I'm excited to have an engineer here on the show. And I feel like that's your primary hat, because you really thought it through in terms of all the challenges, but also how to break through. And clearly that's been your recipe for success so far, which has been tremendous. I mean, you could argue you built 2 careers already, and you got plenty of work left to do for sure. Published hundreds of papers, won a bunch of awards. Most recently, want to highlight it. Congratulations, you won this John C. Polanyi. Sorry for the mispronunciation, maybe Polanyi award from the National Sciences and Engineering Research Council of Canada. And you've got a vibrant and productive lab, which I'm guessing is probably the most gratifying part of it all. But like more and more PIs these days, you've also started a company that's Cuthera, which you alluded to earlier in the talk here in the chat. I'm assuming to bring some of this technology to the marketplace. So for listeners who may be intrigued about maybe moving in that direction themselves, or just curiosity. What are some of the highlights, both in terms of the positive, but also things that to be wary of, perhaps? I don't know, unexpected benefits. I don't know. Break it down for us, what your experience has been like there.
Yeah, so the company that I started for translation of heart on a chip, basically iPS-derived heart on a chip, got acquired, and it used to be called Tara Biosystems. And I co-founded this company with Gordana Vunjak from Columbia University, and Casey Ronaldson, who was in your podcast, and also with 2 back then students in my lab, Boyan Zhang. Now he's a already well-established, very famous professor at McMaster University, and Yimo Zhao, who is a scientist at the Acceleration Consortium at the University of Toronto. So it was a team of 5 of us. We co-founded it in 2014. And basically it was using this like BioWire system that was electrically stimulated. We had a nice paper about it in Cell, and Gordana and Casey had a nice paper about maturation with electrical stimulation in Nature. We also had another paper before that in Nature Methods. And while the company was operational, it grew to about 20 people, and it was like testing drugs for a lot of pharma companies on a service model. But what turned out to be the most valuable asset of the company is human disease models that were built on specific IPS lines. and basically modeling human disease through this approach, right? And the reason for this, some of these phenotypes you really cannot capture in a mouse. Eventually, because of this asset, like human tissue biology, mimicking human disease in a dish, Tata got acquired by an AI company called Valo Health. Who would have thought? When we started Tata in 2014, AI wasn't a big deal at all. It was like something that was just kind of in its infancy. Some, students were working on with their PIs basically. But AI companies, they have a lot of molecules, and especially if they're trying to develop a model for a certain human genetic disease, it's very valuable for them to actually have tissue biology, right? Some of their work will still have to be carried out in animal models, but a lot of validation work, kind of, does it work, does it not work, the molecule, after you make it, it's very, after you make the molecule, after the chemists synthesize it, it's very, very useful to be able to test it on a preparation that actually has that genetic mutation that you are kind of targeting the molecule to. And so it got acquired in 2022, and the acquirer basically held the entire team except a couple of C-level people, which is pretty standard. But the entire team was maintained, and it was announced in 2023 that basically Valo got a huge deal with Novo Nordisk for cardiometabolomic drug discovery, right? So you have the component of heart on a chip, AI molecules, and now like a big pharma like Novo Nordisk, who actually is capable of doing clinical trials, working together through potentially really, really big contract. It could go up to 2.7 billion with milestones.
Wow.
So I think, to me, when you think about what success, right? Like the success is people using the technology, being in the hands of somebody who can really advance it, right? Advance the molecules that come out of it. And I think that was a really rewarding part of the journey. Also, people who trained with me and with collaborators like Peter Bax, they became key leaders in Tata, and then also they're very useful and advanced in Valo as well, like Nicole Ferrick and Ruth, for example. So also, through this kind of activity, if you're doing tech transfer, you can create jobs for your trainees as well, because they're really valuable. They know how to do it, right? Nobody else knows how to do it.
Yeah, they're the ones with the hands-on expertise. And I think one of the cool things about Tara, like what you alluded to, is that it was founded in large part by female biomedical engineers, cardiovascular scientists like yourself, Gordana, and Casey. I think that's actually one of the really cool things about this particular subfield, is that there's a number of prominent women scientists like yourself who are represented. We've had Casey on the show, like you mentioned, and her mentor is none other than Gordana, one of the world leaders in bioengineering and organ-on-chip approaches at Columbia. So could you talk a little bit more about the importance of representation in the field, especially with women taking roles of, visible leadership in this area?
Well, I think definitely it's harder for women, right, to start companies, and it's much harder for women to raise money. And that really hasn't changed, right? The CEO of Tata was a woman also, Misty Ushio. My second company that's more focused on regenerative biomaterials, Qtera, is also very heavily women-led. And it's just, there are barriers, right? Still, it's just maybe something subconscious where it's just a lot easier for men to raise a lot of money. Eventually, basically big VC firms and men still hold the power, right? They hold the money. And there is a little bit of distrust. Oh, can you really do it or not? If we give you that much money. But I would say, figure out how much money you need. Maybe you don't need to raise that much, right? That's another way, right? Like maybe you don't really need to raise that much money. How far can you get? What do you really need, right? What do you really need to achieve that next step? Maybe that's what you should be asking yourself. And I'm really hopeful that this will change, right? As there is more and more women in entrepreneurship, there will be more like basically success stories that you can look to, and basically also path for younger women entrepreneurs to kind of see themselves in those roles and to receive mentoring. So maybe I'm too optimistic, but I think the future is bright in this regard.
Well, you've set a very strong precedent. I mean, that success story, your own success story, is an inspiration to all of us, gender aside. But I think particularly it's important, the visibility, as Arun described there, for younger scientists, and for the VCs, right? To recognize that oftentimes, the idea, of course, we all believe this, but you as you described, the unconscious and conscious bias can kind of overwhelm that lofty notion of the idea being preeminent over whomever is presenting it. But your story across the board is an inspiration, and we really appreciate you sharing the science and also charting a path that you've charted in your wake, of course, but also really talking about the way forward. I love your synthesis of the challenges and the pathway for materials in the cell biology and how they're going to be integrated. I think everyone who listens to this interview and to other engineers on the show, they really stirs a lot of ideas. So again, thank you for joining us and contributing to that forum. Before we let you go, we got to get a little more insight from you, more about yourself, in your own maybe personal science history. First question we have for you is, what's the best piece of advice that you've ever been given, either professional or not?
Well, by far the best piece of advice has been given to me by Bob Langer. It has actually been given to all of us. And I was Bob's student, and that's where I met Gordana, who was a PI in Bob's lab and who kind of took me under her wing. But Bob would always tell all of us three things, right? Kind of think big, work on the most impactful problems, right? He would always say it takes the same amount of time to work on something that's maybe not so important as to work on a really huge problem that hasn't been solved and that's like super important for everybody, right? So don't be afraid to pick the hardest problem because it's going to be the most worthwhile. And the second one is obviously work hard, right? You got to work hard. Nothing happens if you don't work hard. And the third one is surround yourself by the best people. And that's really true, right? You want to surround yourself by really the top people and the best people in that area. Whatever you're working on, surround yourself with the best people, with the best students, right? The students are the most important, the trainees for folks in academia like myself. And you want to make sure that you can attract the best students to work with you. So I think those three pieces of advice are really almost like a recipe, or that's the recipe for how to have an effective academic career and an effective research lab.
Yeah, I mean, the proof is in the pudding. There's some wise kernels from Dr. Langer himself. Clearly took his own advice there on number 3, at least, given his neuro tree is very deep and very strong. You got the pedigree that's come out of his lab. You guys are killers. And I think the best thing about that is that you've extended way beyond his imagination. Talking about the biggest problems, I think the thing he'd be the most pleased with is how you've tackled all these problems that a lot of people didn't think had a solution. Finally, what is the biggest misconception about science that you'd like to resolve here on the show today?
Well, it's for folks who don't know us, right? They think we're like Dexter-like people, right? Like you alone in the lab with a lab coat, and you're like a little bit mad scientist, plotting something that is maybe not to be trusted. But people underestimate how science depends on people, right? It's basically all about communication. Not all, but a lot of it. You cannot do anything alone anymore, right? The time of a one scientist in the lab is over, definitely in stem cell biology. Definitely. Nobody can do anything by themselves. And, having a good network, relying on collaborators, promoting good communication within your team is really critical. So we are very, very social scientists, right? And the importance of that interaction and how big of a network we have. I think then people, when they learn, oh, you're a scientist, but how come you travel so much? How come you know so many people? How come you're always in a conference? It's like, because we have to. You cannot do anything by yourself anymore, right? So I think that's one of the misconceptions that's still out there, that you're like one little bit mad scientist in the lab, kind of working on something by yourself, but that doesn't happen anymore.
Yeah, that's an old idea. And I'd like to say that we've made a bit of progress here on this show, introducing the world to just how dynamic the personalities and the great breadth of talent and diversity that is present in not just stem cells, but in the wider scientific field. So yeah, I mean, as they say, it takes two to think, much less to coordinate the effort that it's going to take to get these cells to work together toward our shared therapeutic endpoints. So that's a nice final message to leave our listeners with. Milica, thank you so much again for joining us and sharing these kernels.
Wonderful. Thank you so much, Dalen and Arun. And I'm looking forward to listening to this episode. Thanks so much for having me, and thanks to Stem Cell Technologies as well.
All right, you guys, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. You can also reach out to us on X at Stem Cell Podcast or by email at info@stemcellpodcast.com with feedback or to suggest guests. I want to hear your feedback, guys, on organ-specific spheroids. It's a hot take. You could tell me what you think. We got another episode coming at you in a couple weeks. Thank you so much for listening.